So let me just give a quick introduction to Jonathan, who I know very well for many years now. He's a great scientist and has been working on various evolutionary domestication issues throughout much of his career. He's now a research assistant professor here at NYU Abu Dhabi. He got his PhD from the University of California in San Diego, and did a postdoc with Michael, and now he's really focused on date palm and the domestication and evolutionary trajectories of date palm. He's published 8 articles, as far as I can tell, on date palm, and that's his focus now. Previously, he had worked on rice and rice domestication. He's published a total of about 40 articles, and many of them, as I said, now focused on date palm, and that's what he's going to talk to us about today. Fruit evolution, and I'll let Jonathan take it over.
Thanks, Ken. Okay,
Okay,
right. So, can everybody hear me okay?
I think so.
Okay, good. Um, yeah, welcome everybody. It's great to see people here from KAUST and elsewhere here in the UAE. And I'm really excited about this opportunity to talk to you guys about some of the work that we've been doing here at NYU Abu Dhabi for about 10 years now. And I thought I'd just begin by introducing you basically to the team, the core members of the team. Of course, we have extended collaborators that I'll recognize at the end of the talk. But I did want to mention Michael Peruganin, who's the PI of the laboratory here and the PI of the lab in New York, who's been a great supporter, collaborator, friend for a long time. And I really, owe him so much, both scientifically and personally, for everything he's done for me. I'd also thought I'd like to mention Dr. Coletta Miri, who's a great collaborator here in the UAE at the Khalifa Center for Genetic Engineering and Biotechnology. And then, of course, our core team members here at NYUAD, we have Tiago, Muriel, now at IRD in France, and we have Sylvie, who recently left us for TII here in Abu Dhabi, and Colette Hazuri, who helped initiate a lot of the work that I'll talk about today. And he's now was a postdoc in Michael's lab here in Abu Dhabi. It's now a research scientist at KCGB. So I think that I just thought I'd start sort of at a high level with the types of questions that we've been working on for some time now. Of course, Michael's group has a core interest in the origin and spread of crops, domestication process that includes both population genetics and questions about the genetic architecture of crop traits, how they evolve, and the types of evolutionary genetic forces that are working on those traits. I'll be talking mostly today in this area with a specific fruit trait that we've been working on, which is fruit color. But the more and more that I've been here in the region, and where I think the climate challenges are sort of at the forefront, the more and more I've been thinking about how, our evolutionary work, parallels very closely because we work on an important crop here. I've been thinking more and more about applications of our work and how we can solve problems or help to solve problems in the date palm industry. So I'll mention a couple of areas towards the end of my talk where I think our work is applicable in that domain. So everyone here, I have to explain these things in the United States because most people don't even know in the US that a date fruit actually looks like an apple, a small apple at some early stage of fruit development. And everyone here in the region also is very familiar with this trait variation, both in sort of fruit quality traits, including the color, the taste, the texture, and other traits that give the crop really an enormous amount of diversity and something that everyone in this region, I think, appreciates and knows very well. I will say one point that as an American, most people don't really know what dates are. They think of them as a dried fruit. It's something you cut up and put in your cereal, perhaps, but otherwise don't really even know. But one thing about my personal story is that my grandfather used to he all the time, every morning, he gave us medjool dates. Me and my brother would each get one medjool date every single morning because my grandfather had a love of dates. So I come with some credibility, even as a somewhat naive American, when it comes to the diversity of dates. Now, of course, the fruit color variation is sort of shown on this slide here. These are fruits that I worked with Robert Kruger to photograph some years ago. Robert Kruger is also a collaborator at the USDA, at the CVARS, the Coachella Valley Agricultural Research Station in Southern California near Palm Springs. And it's a wonderful collection where they have both a lot of cultivars from really North Africa and ones from this region. They also have a lot of the crop wild relatives, which are one of the few locations in the world where you can study the crop wild relatives for date palm. But so the colors variations extraordinary, and an obvious question for people in our lab is, how does this type of diversity evolve? What's the genetic basis of this diversity? And from the evolutionary perspective, originally when we first started working on color, we saw it from in an evolutionary context, and it raised sort of some questions. When you read about the crop wild relatives of the cultivated date palm, this is our best guess right now about what the three closest relatives to cultivated date palm are. My colleague Muriel Gross-Balthasar is working on a robust phylogeny for the genus, which we still don't have because the genus is hybridizes a lot and it creates problems for phylogeny construction. It's always been a puzzle among the systematists. Basically, how is date palm related to other members of the genus? But so the puzzle is essentially that these closest wild relatives all have yellow fruit, and then there was an explosion sort of diversity in fruit colors in the cultivated date palm. There are other, I just put others here. There's 13 or 14 species total in the genus, depending on who you ask. But there are other red fruited species in the genus. And so the capability to make red fruits has been there at some point in the history of the genus, but the ability to make red fruits appear to have been lost and then regained again here in cultivated date palm. So we're interested in that problem. So when we first, so I'm going to show you, basically tell you a brief story of the history of how we have gone about studying this trait. Back in 2014, the first study to map a pigmentation trait in a palm was done by a group of researchers in Malaysia who used QTL mapping. They had a mapping population that dated back to the 1970s that they were able to use to do a QTL mapping study and arrive at a gene which they called fluorescens, because it's kind of the mutant phenotype for an oil palm, which most fruits are of the red type in oil palm. And so they were able to map this trait using a QTL strategy and arrived at this R2-R3 MIB transcription factor, which is a known activator of anthocyanin pathway. So this MIB is a ortholog to PAP1 in Arabidopsis, which is a subgroup 6 R2R3 MIB. And they found a series, essentially an allelic series, a set of truncated alleles at this transcription factor that each of them in the homozygous states leads to yellow fruit. So that paper came out about, 3 weeks before we were ready to submit a paper in, actually it was maybe 3 months. But we were about, actually, no, we were going to submit it, and the work just came out. I think it's 3 weeks, if I remember correctly. I mean, it was very close to the time where we were looking to submit. And we decided, hey, let's take a quick look at this gene just in date palms to see if there could be something interesting going on. And what we found was that yellow fruited varieties had a transposable element insertion in exon 3 of this ortholog to the oil palm fluorescence gene. And, it became pretty clear pretty quick that this was likely to be a functional variant that controls fruit color variation in date palms. And so we just did and reported these figures here in this 2015 paper, basically showing that there's a pretty strong association between genotypes defined by the presence or absence of this transposable element insertion in exon 3. And so I put this here to recognize that Michael decided to give this insertion a name, and he decided we should call it a binmijid after the navigator or explorer from this region who explored the region and developed maps in the region dating back to the 15th century, to the 1400s here. And so we'll call this, throughout my talk, we'll call this a binmijid, and I'll use this notation here for Vir plus or wild type allele, which is a fully functional set of 3 exons, and the binmijid insertion allele, which we call the IM allele. So fast forward a few years, Dr. Colette Hazuri really took leadership on this project and developed the first GWAS mapping population for date palms. And it really consists of 2 existing repositories, one a research center here in the UAE, and a second private farm from a private collector who collected a lot of date palm varieties. And so together, there's about 120, there are exactly 127 varieties in this mapping population, some of which we have replicates for. And so we were able to basically map the trait and found that the same gene that we had suspected was the predominant or primary QTL for this trait was mapped to chromosome 4, and so we're pretty sure that this is the most important QTL for this color polymorphism. So just give you a few more details here. One thing we noticed was that there are some varieties that have yellow fruits that do not sort of match the predominant color that we would expect from a wild type homozygote. And so we looked into that in more detail, obviously, to try to understand what was going on there. And that led to the discovery of another allele that we think is an important factor controlling the trait. And this is a start codon mutation that codes for ATA instead of the canonical ATG. And all of the fruits that are homozygous for that allele are yellow. So we've seen this, the allele is actually at pretty high frequency, so it's not uncommon to find homozygotes for this what we call MET minus allele. And the allele also acts, as you might expect, if it's a loss of function, as fully recessive to the wild type allele. So we see heterozygotes that are always red when they are paired with a wild type allele in this met minus allele. So this, we think, is the predominant locus that controls food color variation, and we can see that if we just calculate the percentage of variation explained, that it explains about 2/3 of the variation in color. But you can also see that there's some interesting genetics here, particularly in the heterozygotes for the abinmigid and the wild type allele. So this combination here leads to kind of a diversity of colors. And so most of the intermediate colored fruits, and some of these are familiar. This is Deglet Noor, which is a heterozygote. Medjool is a heterozygote. Bentamota, this is an Egyptian and Sudanese variety. Saidi, this is an Egyptian variety. These are all heterozygotes, and there's a lot of diversity. The full range of diversity, in fact, in fruit color variation shows up in these heterozygotes. And that means that there's some factors here that we don't fully understand. And I'll probably not go into that too much, but we do see what they saw in oil palm. So oil palm, they described it as a dominant negative mutation, these truncation alleles, they believe, in the heterozygous state. These are heterozygotes for wild type, and the truncation allele are yellow in oil palm. And we see that here as well in a large class, these varieties. And we think that, I mean, this R2, R3 MIBs form dimers in, to work as functional transcription factors. And there's a lot of work that's been done on dimers. In fact, C1 R2, R3 MIB from corn was the first R2, R3 MIB to be studied in any detail. And it's the sort of quintessential example of dominant negative mutations in plants. And so these are cases where we think that something about the dimerization process, and there's a number of models for this, but something about the way that the dimers form cause the transcription factor to essentially not work when one of the alleles is truncated. And so you end up getting this dominant negative effect. So fruit color then in date palms, the objective, one of our objectives was to try to understand how it evolved. And at least now we have some idea about the genetic basis of the trait in cultivated date palms. And so we were interested in going a little bit further to try to understand this sort of interesting curiosity for how, if you lose a pigment, how do you go about gaining it back in this case? And so one thing that I just started to do was to look at the sort of the haplotype diversity for the different alleles. So we did use these data here come from allele-specific PCR. So we've taken heterozygotes and sequenced the gene separately for each of the two alleles in heterozygotes. So we have full haplotypes. So these are not phase data. These are actually inferred haplotype sequences for this gene. And if we build a sort of haplotype tree, we can see that the first thing to notice is that there's a pretty deep division between sort of the wild type class and the abinmigid type class haplotypes when we rooted on oil palm. And second thing, I guess, to notice is that this Vir-Met minus allele is sort of derived. There's no genetic variation in this haplotype of this gene. And I won't show additional evidence today, but we think that this is a very recent allele. However, the wild type and the abinmigid alleles are quite old. And part of the additional evidence that they're old is by sequencing, again, haplotypes in the wild relatives. And what you can see are that the two closest wild relatives of date palm, in the case of Phoenix theophrasti, share an exact haplotype with these are North African varieties, and I won't be talking about it today, but some of you may be familiar with our work showing admixture history of the date palm crop, where we've seen very strong signatures of introgression or admixture between Phoenix theophrasti, this Cretan date palm, which is a distinct species, very different genome, and yet we see distinct signatures of shared ancestry with cultivated date palm in the recent, what we think is in the recent past. But we can see here the complete sharing. This is actually Medjool and Deglet Noor, the two predominantly the largest commercial cultivars in North Africa. And Phoenix theophrasti has an identical haplotype with those two, likely, we think, due to introgression of this allele into the cultivated date palm. But we also see Phoenix sylvestris. This is the Indian subcontinent wild relative, which has also is segregating for this abidmejid type allele. But it's also segregating for what looks like a wild type allele. So this Phoenix sylvestris has both alleles, as does cultivated date palm, therefore possibly suggesting that this this wild type class of allele was vertically transmitted potentially during evolution from a common ancestor with Phoenix sylvestris. And of course, the abidmejid allele may also have been vertically transmitted over evolution from this common ancestor. Now, of course, Phoenix sylvestris has yellow fruits. I'm coloring it here red just to indicate that it's a wild type sort of allele in this clade. But we think that there's secondary mutations that have accumulated in Phoenix sylvestris such that the fruits are always yellow. And that's an area of interest of ours to understand what controls fruit color, not just in cultivated date palm, but in other members of the genus. So I'll make one final point here. In some preliminary data, we have seen the abimejid allele in relatives as divergent as Phoenix reclinata. This is a sub-Saharan African species, which we believe is significantly more diverged than the closest wild relatives of cultivated date palm, which include Canariensis sylvestris and Theophrasti. I'm just showing you here a wild type copy that is identical to Phoenix canariensis in this case. But we think that this allele, this abidmejid allele, has been around for a long time, which was somewhat surprising initially. I mean, what we kind of expected was that, that maybe this insertion was very recent, maybe even occurring during the domestication of cultivated date palm, but in fact, it's actually quite old. So here's just kind of summarizing that in sort of the visual case where we can look at the allele frequencies of these sort of three sort of alleles, and we can see that Phoenix theophrasti is in fact fixed for this abidmejid allele. The two alleles, the wild type and the abidmejid, are segregating in the Phoenix sylvestris. This shows you that within the cultivated date palm, there's a lot of these three alleles are all at pretty high frequency. And so we see them in various combinations at high frequency. We've published on these ancient date palms, these Judean date palms that were germinated from seeds that date back to 2,000 plus years ago. They have all three alleles in that Judean population. And Phoenix canariensis has yellow fruits, of course, but also is fixed for a wild type allele. So we think that there's a second independent causes of the yellow color of Phoenix canariensis. So some of you are probably familiar with LTR retrotransposons and their insertion mechanism. And evolutionary biologists capitalize on the fact that when LTR retrotransposons first move and they insert in the genome, the sister LTRs are identical at that point, and they must be because of the mechanism of insertion. And so we can date this LTR retrotransposon insertion by assuming a mutation rate, essentially in a molecular clock. But the first observation is just that the LTRs are divergent from each other. So what I'm showing you is an Iraqi varieties sort of sequence here. So again, we've used haplotype specific sequencing to sequence the two ends of the transposable element from one allele in one variety. And I'm showing you an alignment of those two sequences. And what you can see is just how diverged they are. They're about 5% diverged from each other, which indicates that this haplotype, that this insertion rather, is old, consistent with the sharing of this insertion with the crop wild relatives of date palm. One thing that surprised us a little bit is we worked with Shojun Wu, who is the developer of LTR Retriever, which is a nice, very nice software to characterize LTR retrotransposons in plant genomes. And he helped us just to, with this analysis, to demonstrate that not only is abinmigid old, it's in the top 2% of oldest transposable elements in the cultivated date palm genome. And that's somewhat surprising for a few reasons, but perhaps the most surprising thing is that it's also polymorphic, right? Most of these are fixed transposable element insertions in the date palm genome, not polymorphic within the cultivated crop. And so we have an ancient polymorphic transposable element insertion that we believe is the primary causal variant for fruit color. And if we apply a very conservative mutation rate, the insertion time is at least 2 million years old, and this is an extremely conservative mutation rate, and so we think that it's likely to be much older than this. So I'm more or less going to leave you with that. I mean, what can we say about fruit color evolution and this curiosity with respect to the diversity and the explosion of diversity of fruit color in date palms? I don't know the answer right now. What we know is that that the abinmigid allele is very old. It's found in these two species here. It is not found in this species here. And so we think that allele has been around for a very long time, but it doesn't really explain where did the wild type allele come from. We see what looks like a wild type allele. We have not seen the exact same sequence in Phoenix sylvestris. So it could have been sort of vertically transmitted, and maybe there are fixed secondary mutations in sylvestris in that species that cause the fruits always to be yellow, and they somehow are maintaining this, or at least the common ancestor maintained a functional allele, even though the common ancestor was likely yellow. And so perhaps the allele was just sort of inherited, and then selection perhaps by early cultivated date palm farmers sort of led to this trait on sort of standing variation, selection sort of on standing variation that already existed. But we cannot yet reject the idea and that this trait came from hybridization with more distant wild relatives. So there are a number of wild relatives that have red fruits. And one possibility is that introgression, because of the promiscuity of these species, that introgression of this trait or this allele could have come from a wild relative that has red fruits. And so that's something that we're working on right now. Okay, so I'm just going to tell you about a few of the other things that I've been working on and sort of directions that we're going right now. Of course, we're continuing to work on fruit traits, continuing to do more mapping on other fruit quality traits. That's one of our core areas of interest. But I've also, in getting to know the date palm literature a little bit, realized that there's not really been a model proposed for how fruits develop in any kind of detail. And so I've been working on synthesizing data from the literature, combining that sort of synthesis of the literature with our own gene expression profiling data. So what we've done is to collect gene expression data and from different varieties from 5 stages of development, and look to see how gene expression changes during fruit development to try to understand sort of the basis for the physiological changes that take place. And I'm working to sort of build a model for what exact genes are likely to be controlling those changes in the physiology during development. And hopefully, once we have that, we'll be able to identify sort of key genes that are playing key roles both across all varieties, and also identify genes that are playing key roles in giving individual varieties sort of their characteristic fruit qualities. And I'll just give you sort of one sort of tantalizing piece of data here that in fruit crops, as you know, the photosynthate that is fueling the growth of the fruit and the expansion of cells, the growing of the fruit, the photosynthate is coming from the leaves. It's being transported to the fruit, and so one of the questions for how fruits expand like they do, that's sort of central in fruit development, is how do they create this sink for sucrose? So sucrose is the transport sugar in date palms, and it's coming into the fruit, but there needs to be a sink there so that the sort of gradient or in concentration continues to pull sort of the sucrose into the fruit as it develops. And so there's a few sort of genes that could be controlling this, and this particular pattern of gene expression suggests that the gene that is probably controlling that in date palm is sucrose synthase. And there are 9 sucrose synthases in the cultivated date palm genome, but there's only one of them that shows this pattern of a strong or very high expression early in fruit development. So I'm showing you 5 different varieties here that all show more or less the same pattern, which high expression early in development, followed by, by the time they're at the late Bisser stage, they're, this gene is essentially not active anymore. But so early expression, and this consequence of this is that sucrose is being sort of pulled into a larger sort of carbohydrate molecules, together with sort of, you can see the pathway here, that sucrose synthase leads to sort of this UDPG and on into cellulose and other carbohydrates. And so this could be the gene that's really responsible for sort of the establishment of the sucrose gradient in date palms. So we have a lot of sort of anecdotal stories that we can tell by looking at the expression of individual genes. There's more we can tell by looking at gene regulatory networks. And so we're using these data to build those networks and understand essentially how the fruit develops from genomics perspective. So another sort of tantalizing piece of data that comes out of this expression profiling was essentially doing differential expression now between varieties and looking at differential expression in between red and yellow fruits. And so we know that the red color is in the skin of date fruits is an anthocyanin pigment. And so there must, we think that the Beresins R2, R3 MIB, it is a subgroup 6 MIB related to PAP1. These are well known to activate genes at the lower end of the anthocyanin, of the flavonoid pathway, I should say. And really, we had a few candidate genes ahead of time that we thought that virescins might activate. And those genes that it probably, based upon just comparative work or comparative studies, this MIB typically targets one of these three enzymes at the lower end of this is what I'm showing you here is sort of the flavonoid plus this is the phenylpropanoid sort of pathway here, and flavonoids are sort of down here. But what we can see is if we just now look at the relative expression between kinesi, or a common red variety here in the UAE, and Nebat saif, also common yellow variety, and we do differential gene expression then between them across these 5 stages of development, what we see is that there's a huge difference in gene expression at 120 days post-pollination. This is when the fruit is first taking on its yellow or red color. And so what we see is a more than a 20-fold difference in expression of this gene UFGT in the sort of late stages of development before the fruit starts to ripen. So they're close to physiological maturity here where they're taking on that skin color. And what we can also see is that this is actually the most differentially expressed gene in the genome between red and yellow varieties. And we're pretty sure that that means that virescins, when it's non-functional in something like Nebat saif, is essentially not that the transcription factor cannot activate UFGT, and it leads to a massive difference in gene expression, which we can see here. And we think that this is essentially explains sort of at the biochemical level why we have yellow fruits, is because the transcription factor that activates anthocyanin is not working, and that creates this difference in gene expression. So one other area that I've recently taken an interest in, and this is more on the applied side of things, is the problem of simulclonal variation. And probably most of you here know that the way that the date palm crop is propagated, at least in large scale production contexts, is using plant tissue culture, and somatic embryogenesis is the predominant technique that's used to produce these clonal plants. And the problem with somatic embryogenesis is that it occasionally creates off types. And there's a lot of evidence in the literature that these off types are epigenomic in nature, that there's some ability of the plants to revert from some abnormal growth phenotype back to normal growth habits later. If farmers leave them for 15 to 20 years, there's well-documented examples in the literature showing this reversion from some off-type phenotype back to some normal phenotype. This is one of the problems here that that's relatively common. This is a Colossus plant that I sampled here locally recently. It's an off-type for what's we call a supernumerary carpal trait. And I meant to put the arrows. So this is from one flower. Typically in date palms have 3 carpels in the female flower, and 2 of them are suppressed, leading to 1 fruit that's produced per flower after pollination. But what you can see here that the loss of suppression of the carpel of those 2 carpels, and the retention of this is kind of like, I think of these as like a vestigial tail in a whale. These staminodes are primordial sort of male parts. So you can see here, I meant to label it with the arrows here, but this is a staminode, this is a staminode, and this is a staminode. So these are just sort of, because date palms are dioecious, I mean, this is, and they've been, dioecious is an ancient trait in this clade. And so it's, these are essentially caused by lack of suppression of male genes in the female flower that are leading to this sort of development of this sort of complicated looking fruit. And so one thing that I've been trying to do is start to work with farmers locally and producers to try to quantify the scale of the somoclonal variation problems, um, because it's a sustainability issue, and I believe it's a major one, because in talking to the largest producers here in the UAE who are planting date palms on the scales of millions of plants, the somoclonal problem is about 10% of the plants. Traits like this one are not apparent in the plant until about 4 to 6 years when the plant first produces its first flowers. And here locally, farmers are watering these plants, at least in the summer, with anywhere from a minimum of 100 liters of water per day. But the common sort of wisdom amongst the agricultural the officials in the ministry here that are working directly with farmers is that farmers overwater their trees. So they're putting as much as 1,000 liters of waters on their per day per tree on these plants. And so if you do that for 4 to 6 years before you even realize that there's a problem, we're talking about a massive amount of irrigation water that's completely lost because these trees with these somoclonal problems are completely unproductive. And the farmers, when they detect the problems, have to take them out. And this particular farm we visited, it's a high variance. So some farms have a huge percentage of the plants are affected. So this one, 90% of the colossus trees on this farm have this problem. And so the farmer planted 400 about 6 years ago, and then realized that 90% of those trees have this problem. And the farmer's putting 200 liters of water per day on these trees. And you can, I didn't do the math for you here, but there's 16 million productive trees in the UAE, we think. And, if 10% of those have had the somoclonal variation problem, and people water their trees with extraordinary amounts of irrigation water for long periods of time, it's a major issue. And so we're thinking about ways that maybe our genomic technologies can be used to understand these traits, to understand what exactly is causing the problem. I think what our idea is we're not going to solve this somoclonal problem in the sense of changing formulae that are used in tissue culture to produce healthier plants. That's a pretty resource intensive endeavor, but we can make a marker for it. I think. That's sort of our idea, is that we could pre-screen plants after they come out of the tissue culture lab and identify the epigenomic change that is causing the problem, and then basically have a marker that we could use to filter out the plants that are going to be unproductive after some time. So this is something we've just sort of started working on, and I'm hoping we can convince one of the local ministries that we might be onto something here, and that they might fund us to work on this further. So also on the applied sort of side of things, we've developed markers for traits, and so it makes sense to find opportunities to use these markers in breeding contexts. And so there's a number of breeding programs going on around the world. And so, we started with our fruit color marker. We believe that we can take a small plant, a small seedling plant, and at least predict whether or not, yes or no, will the fruit produce anthocyanin pigment in the skin. And so there's a strong interest in a number of countries around the world to produce red fruits at the Bisser stage. And so this is something that I think we can contribute. So throughout our sort of studies of the genetic basis of fruit quality traits, we're looking to develop markers that can be used by breeders in that context. Okay, that's really it for for my presentation today. I just, again, wanted to thank core team members, but also extended collaborators, including Rod over at Calst, who's been with us for a long time, working with us. I would also say Frederique Aberlenc was important and instrumental early on. She's at IRD in France. And then Dr. Ahmed Al-Falasi is the private landowner who so graciously opened up his farm for studying diversity of date palms. And so we couldn't have done it without him. So thanks a lot. I'll open it up and for questions, I guess, Alia. Is that okay? Okay.
Yeah, great. Anybody have questions? You can raise a hand or just chime in.
Yeah. Hi, Jonathan. Hi, Kenneth. I would have a question. Sorry, I arrived a bit late. I couldn't make it earlier, but maybe I missed a point. But did you look at the methylation pattern of your T insertion that is related to fruit color? Because you were looking at, I was thinking when you were talking about this somatic clonal variation, that is very interesting to look at epigenomic variation. But did you look also at the between variation?
It's a great question because I think a lot of the diversity in the fruit color, particularly in the heterozygotes for the abinbegit and the wild type, could be epigenetic. I mean, it could be second site variation, sort of another gene that's sort of acting as a modifier. But my hunch, and I think probably what you're getting at, is that it could be epigenetic. And there's a number of mechanisms that, but unfortunately, I haven't. It's a hard experiment. I've collected some of the fruits that I need to do the experiment, but it's a bit of a tricky experiment, just to dissect the skin and to do the RNA-seq on it, and the methylation, on the individual skins material. But anyway, yes, I haven't done that yet, but definitely on our to-do list.
Oh, cool. And just a question about that. For dissecting and so on, is there any possibility to use a single epigenomic in that case if it's too complex to dissect different parts? Or it's not something that you can think about on date palm? And
I've actually, Ken will be interested to know this. We've put in a proposal with Sunil to see if we can start this. So a former postdoc of Ken's is really a world's expert in getting single cell to work on different cell types and different tissue types. And so we've been talking for years about trying to get that to work on date fruits. And so I'm gonna try to give that a try if we can get some money to do it.
thanks a lot. Thanks a lot, Doc. Nice
Sure.
talk.
Thank you.
Jonathan, I have a question on the somoclonal variation. It's a huge problem all over the place, right? And
Yeah.
and this seems I'm really curious about this farmer with 90% of his trees being aberrant in some way. So one question is, are they all aberrant in the same way? I don't know how much this is answerable, how much this was just a survey. And then, yeah, so maybe that's the first question.
Okay. Yeah. I mean, on this farm, all of them have the same problem.
The exact same problem.
Exact same problem. I went tree to tree to tree, and they all have this trait.
I would be curious about
Yeah.
his method or his source of the
Right.
propagules, of not propagules, I guess, but the
Yeah.
clones. Are they from the same, because the way this is done is from, all the way back to callus, right? And so is this coming from this, are the trees themselves
So,
clones? Well, I guess everything's a clone.
they're clones. They're the same variety, and they come from the same tissue culture producer. Now, I'm careful. This is a sensitive issue in the industry, and I was nervous about starting to talk about this both with the tissue culture companies and also with the farmers. People are broadly supportive, but what I do make sure to tell and want everybody here to know, in the event that you have friends or interests in the tissue culture community, is that I tell the farmers, first thing I say to them, I don't want to know who produced your trees. Because, you know, it's a sensitive issue because the tissue culture companies are aware of the issue, but they would rather the issue goes away. The farmers, of course, would also rather it just goes away, but they're also having to bear brunt of the costs of it. And so I just make sure that everybody knows that we're not interested in pointing fingers. That's not my purpose. But we are interested in you know, finding a solution to it. And so, but yeah, in this case, I do ask, are they from the same source? And in this case, yes, they're from the same source, and 90% of them had the same problem.
Yeah, I'd just be interested, you know, I'll kind of leave it at this because I know there's probably not, you know, this is all information that's not probably collected, but it'd be interesting to know if the same aberrant traits pop up from different sources, because then
Yeah.
as far as your goal finding markers, you may have specific regions of the genome that are susceptible.
Yeah, that's definitely the goal. And I can tell you it's definitely conventional wisdom that the same problems can crop up. I mean, so independent of, you know, what we've been doing to try to figure out where the problems are, the same problems do pop up from from different labs. And they can, you know, this was an extreme example. I mean, it's one of the ways we were able to find the farmer who had the problem is because, you know, asking a few people, and we arrived at his farm. But we see low-level cases also. One guy that I spoke with recently had 2 trees on his farm, and he has had them there for 15 years, but he kind of keeps them around as a curiosity, you know, on his farm. But same problem. I'm not actually in that case certain that it's from the same laboratory, but the problem is across the region, right? It's from, you know, I know farms in Morocco that have the same problem, and these are going to come from different tissue culture laboratories. So our goal right now, I mean, and if anybody here, you know, has farms or knows the industry or knows people, you know, we're trying to get the word out. We don't want to really, you know, point fingers at the tissue culture community, but we do need materials to study, right? There's no research germplasm repository where you can go and study these problems. And so we have to find them where they are. We also have to, you know, convince farmers to not root out the trees, you know, because we need to study them for a couple of years. And so, but if anybody, you know, has friends of friends or whatever, and, you know, we're definitely looking for more opportunities to find trees. We have enough to do a small study, but we would like to know more. And we'd also like to collect more information. So we're starting a database to basically try to find out how much generally the somoclonal problem is, right? I mean, there's a few papers out in the literature that you know, quantify that at least locally, but in the UAE, no studies of this. I don't know of any in Saudi either, that have been sort of systematic in going through and finding out just how big the problem is, so that we can really make a projection for just how much water is being wasted because of this.
Yeah.
Can I add something on? Oh,
Yeah.
sorry, Ken. No, go ahead, Ken. Or
No. I was going to open it up. Go ahead.
yeah. It's true. It's a real problem. The somoclonal variation. There are a lot of farmers that are buying plant issues from the tissue culture, and then after 15 years, they found out that they cannot produce flowers, or they produce flowers that are not fertile. And I think what is done is done. I don't think we can cure the problem. We can identify it. But what probably the tissue culture community, what they should do is go into genomics. I mean, I think Jonathan is starting this now, and then provide a certificate for every farmer. Okay, your plant is by doing genomics, and this plant is free from mutations. I think that's a step that's it needs to be done before moving forward with the tissue culture and then and tissue and propagation via tissue culture. Otherwise, it will not be sustainable. There are a lot of farms here, so Jonathan, if you want contact, I can put you in touch with some people, especially farmers. They were really willing to share their material. They are not pointing any fingers on anything. They just want to know what's the problem and how can it be
That would be wonderful. Thank you so much for
Yeah.
the comment you made is right on point, and I would greatly appreciate help with.
Hi,
Yeah, and
thanks.
epigenetic is something that is, I think it's not only going through fruits, but it's something is altering the whole date palm
Yeah.
life cycle, starting from germination to callus propagation to flowering to male, female, gender determination to fruit formation. It's really highly epigenetically controlled.
Yeah, I think that until we really get some studies sort of on this from the epigenomic side, I don't think I don't know if my thinking is that we're going to need a marker for each of the different traits, as opposed to sort of, if you had an epigenomic profile, that's probably pretty cost prohibitive, I would guess, on the tissue culture side to screen plants, to at least right now. But I also don't know if, though, we can tell, just from the total epigenomic profile, if this is going to be a problematic plant or this is going to be a problematic plant. I mean, from other crops, what it looks like is that the epigenome is sort of completely messed up in these plants that show these somoclonal variants. And, so you have a normal coloss, and then you have a coloss that has the somoclonal variant. Like, I'm expecting when we get some data on this, that to see sort of the entire profile is sort of changed in the two plants. And so you don't really know which one is the problematic variant. Perhaps there could be a generic marker that could tell you, here's the points that the places in the genome that are always sort of altered in somoclonal variant plants. And maybe there could be a generic marker. So that would be the ideal situation if we could, we wouldn't need to understand, this particular supernumerary carpal and staminode trait, and we wouldn't need to understand dwarfism. We wouldn't need to understand, other vegetative traits that show up, or the lack of fruit set of any kind, pollination problems, this type of thing. It'd be great to have a generic marker, but the biology is gonna tell us whether or not
That would be a very wishful
thing. I agree.
Yes, that's going to be very difficult.
Yeah.
Hi,
Okay, I think we had another question. Yeah, go ahead.
Hi, Jonathan. Thanks for the great talk. I was wondering, is there any correlation between the evolutionary link of different palm trees, especially in chloroplast genome, and their adaptability, especially?
I mean, I think I think conventional wisdom is that the chloroplast genome, it has a very slow mutation rate. There's not a lot of diversity. So I don't think the chloroplast is affecting sort of adaptation too much. People have been using the chloroplast as a marker in this genus for a while to try to understand the evolution of the genus. So you can find probably 5 or 10 studies where people have sequenced either all or part of the chloroplast genome and tried to reconstruct the phylogeny from it. And those are interesting papers, and I think we've learned a lot from them. But, in this genus, the problem is that the genus is very promiscuous in the sense that the different species hybridize a lot. And so the problem with building a phylogeny and really understanding the evolution of the genus using a conventional molecular systematics approach is that different genes in the genome show different relationships between the species. And because of that, it's hard to figure out what the correct sort of phylogeny is, because of this, movement of genes or molecules like the chloroplast across species boundaries. And so, we've seen, very peculiar relationships in the chloroplast genome, things that we didn't expect. For example, that the North African cultivated date palm has a chloroplast genome that's more closely related to Phoenix sylvestris than it is to Arabian varieties here in what we sometimes call the Eastern germplasm. There's this weird sort of pattern, and we have no idea why that is, but it could have something to do with sort of ancient hybridization. But I hope that answered your question.
Thank you.
Okay.
Yeah, someone is in the chat menu here.
Chat, yeah. Do you want to? Okay. No bearing fruit.
Yeah. Malabab is saying that not bearing fruits is likely to improve as the tree grows, and that's true, right? I think he's referring to the reversion due to the epigenetic factor. I think what he's commenting on, and that would certainly fit with what's been published on the problem. Which is why the predominant reason why we believe strongly that these traits are epigenetic and not caused by actual mutations.
Okay. All right. Any other questions on any of those topics? We could take a few minutes over if anybody does. Okay, if not, thank you very much, Jonathan. That was a stimulating and really interesting talk, and perhaps even fruitful in another sense of the word for some of the data you're trying to collect. So great, and it's great to see everybody. Thank you for joining. Stay tuned for more, hands clap. Stay tuned for more topics, and audio will send out the emails on the various speakers who are coming up, and it'll be great to see you then. Okay. So long, everybody.
Thank you, Ken.
Thank you, everyone,
Thank you,
and
John.
have a great week and great month and
Thank you, Ikram.
pretty Christmas.
You
See
too.
you.
Too, Ikram.
Bye-bye.
Bye-bye.